Thank you so much for joining us today for a special memorial session in honor of the life and legacy of Philip Benfey, who passed away in fall of last year. I'm Rachel Shahan. I was a postdoc in Philip's lab from 2018 up until February of this year, when I left Duke to start my own lab at UCLA. And it's really been a pleasure to organize this session together with Wolfgang Busch, who was also a former postdoc in Philip's lab. So in my time of knowing Philip, I came to know him as really a creative and inspiring and forward-thinking scientist. So today you're first going to hear 4 short talks talking about Philip's contributions to plant development, to genomics, to systems biology, and also his accomplishments as an entrepreneur. Then after that, you're going to hear short tributes from 10 former trainees who are going to talk about Philip's phenomenal role as a mentor in their lives, and also to give a little bit of a personal feel for what it was like to work with Philip and really get to know him. So on that note, I would just very briefly like to say, one of the most important things to me about working in Philip's lab was the sense of community that he was so amazing at creating. And he created the sense of community by being very clear that he cared very deeply for us, not only as scientists and our growth as scientists, but also as people and our growth as human beings. So in light of that, we really got to see sometimes a softer side of Philip. So this included when he would give the lab grandkids rides in his gator at his house, and when he would really humor us, he wore the lab birthday crown and allowed us to sing happy birthday to him while he conducted us. And just getting to see those parts of Philip and getting to spend that time with him was really important to me, and something that I'm going to remember as I begin my own lab and am a mentor myself. So the community that Philip created is long-lasting. This is his 65th birthday, which was my very first week in the lab. It was a really special time to get to meet all of his former trainees. So I'm very glad to be part of this community. Philip is deeply missed. He was deeply loved, deeply missed, but I know that his legacy will live on for a very long time. So Wolfgang?
Thanks, Rachel. So it's a great pleasure and really an honor to be together with Rachel organizing this session, and we really hope we can give like a comprehensive overview in the limited time that we have. And maybe I just want to add one more aspect. Philip was this amazing role model, this amazing scientist, this amazing mentor and entrepreneur, and also he had like a really funny side to him, but he was also what I always found like deeply impressing as well, and what I really miss is he was a man of culture. So, okay. There should be a slide. That's also a very nice cultural, rose. So maybe I just tell you the story then without the slide. There was one last slide at this, not that. It was in the first, okay, so it didn't get into the slide deck, okay. So I remember when I met Philip after I left the lab, there was around 8 years after I've left the lab in Oviedo in Spain, and we were spending like 2 days together at this conference and walking around the city. And it was amazing, the stories about the art he had. It was, we kind of posed, and there was this photo at like a famous statue called The Traveler, and he knew all the backstory without reading it up, and it's so impressive. And a little later, I actually found this book from his brother, and it was kind of like an accident, because in that, when we hung out, I asked him about his family, because his great-grandfather was kind of translating the Sanskrit, very important documents from Sanskrit the first time. But he said, "Oh, you read the book of my brother," and I didn't know his brother read a book. And so I really want to recommend you, if you want to know something about the origin of Philip, because in this book there's this wonderful interwoven history about Philip's family when they migrated out of Germany and came to North Carolina. And part of this family was academics, part was artists. And if we want to learn more about Philip and his cultural side, I think you should check out this book by Theodore Benfey. I definitely enjoyed it a lot. Okay, so without further ado, I'd like to introduce the first speaker, Dominique Bergmann from Stanford. And she will talk about the impact of Philip on plant developmental biology, because he really left a big mark. Thank you, Dominique, for coming, and let's get on with it. Hopefully you can get the slide.
Okay, let's see if we can get the next set of slides up. Oh, okay, perfect. So I wanted to first thank Rachel and Wolfgang for inviting me to be part of this tribute to really a truly amazing colleague in plant biology, Philip Benfey. And I think it's really a testament to his impact that we're both opening and closing this meeting with events that honor him in some way. This set of talks right now, and then the mentorship awards that have been named in his honor at the end of this meeting. So when Rachel and Wolfgang asked me to be a part of this, my immediate reaction was, well, of course, Philip deserves to be honored and celebrated and remembered very broadly. And then my second response was apprehension, in part because of the enormity of the task. You know, how do you properly capture and distill in a few moments the essence of someone who did so much and who was so much? And also, it was intimidating in part because, unlike many of the people here, I was not a student or a postdoc of Philip's, nor was I a collaborator, and we've never co-authored a data paper. And I also work on the other side of the plant. So, but I began to really appreciate what Wolfgang and Rachel did by organizing the talks in the way that they did, which is to really emphasize that Philip's legacy does extend to all of us, even those of us at a bit of a distance. And this is perhaps even a greater contribution, that Philip really impacted a much wider swath of science and scientists than those in his direct orbit. So I'm here to talk about Philip's impact on developmental biology. And if I were to summarize it in a single sentence, is that he literally changed the landscape of plant developmental biology. And he did this in several really important ways. So first were some important discoveries, specific discoveries that he made about gene regulation in the service of cell identity and tissue organization. And I'll highlight several of these in the next slides. But second, he was a brilliant presenter. So these discoveries that he made, intrinsically they were interesting, but it was his presentation of these. He was a great communicator in writing and especially in person. He had this resonant voice, his movement across the stage, which I can't do because I don't have a mic here, but he would just, you know, out there, and you all know this. And you know, also his propensity to dress far better than any of the rest of us. So he really compelled people to listen to these insights in biology. So Philip not only had these great ideas and could communicate them, but also recognized that to do these things you need resources. And he was a fabulous advocate in the broader communities for this vision of plant biology and the funding vision for plant biology. And then finally, mentorship. It's been brought up before. It's going to be brought up many, many times after this, which is that Philip's legacy is firmly established and it's feeding forward in the people that he mentored. And really, this is something I've really enjoyed interacting with many of Philip's mentees and their mentees as well. So this is really amazing to see. Okay, so I'm going to talk a little bit about some of those specific discoveries. And this is really challenging because Philip made many fundamental and paradigm shifting and just like, that is so cool discoveries over about 30 years of research. So I'm going to highlight a couple of them. So chronologically, from screens that were done in the 1990s and classical developmental genetics, Philip really defined how transcription factors, or TFs, work to generate cell types and how they pattern those cell types. And summarizing a lot of things here, a few of the papers that were really key for this, and an image from one of them that I think we've all seen pictures of Philip with this image. Let's see if this actually is a pointer. Look at that, it's a pointer. Okay, so, and what was really fascinating about this particular work was that there were transcription factors and they were related to cell identity. And this was broadly known to happen in developmental biology. But this idea that a transcription factor could not only specify a cell identity, but did so in a particular pattern, and there was communication communication between layers in a very, very, very short pathway, that being a transcription factor moves from its site of origin here in the stele out a layer, specified those cells, interacted with its targets and partners, a beautiful set of papers all illustrating how this very simple system could pattern and continue to pattern this root. So this is a really important set of things, and it's continuing and growing. Another thing that, again, in the context, and I'm going to talk a lot about these things in the context of developmental biology as a broad field, of which plant developmental biology is an important part, but typically a small part. And in developmental biology, the idea was to be very rigorous. You needed to isolate things, get down to individual cell types, and that usually meant dissociating cells into a dish, and kind of asking what genes turned on and off in that very artificial situation. And I think this is work that I'm going to show one image actually from Rachel's work that's quite recent, but summarizing a number of other things, where the idea was that one of the things that was hard to do in plants, it was hard to do tissue culture, in fact we still don't have that, but was to recognize that you lose things in doing that. And if you could come up with a way of capturing very detailed genetic information from a recently dissociated organ, you could start to really build in that idea of how cells were behaving in their normal context. And so this kind of idea that to understand gene networks, you couldn't just do tissue culture, but put them back into the organism. It's being rebranded these days. It's this new exciting hot thing. It's 4D physiology. It's multi-scale modeling. And this was all done by Philip many, many years ago. So kind of coming full circle there. And then finally, I wanted to just mention another thing about a lot of looking at gene expression or genetic networks that were happening in these intact organs and organisms, and that a lot of things were cyclical. It didn't just happen once, but it needed to be continuous, and there are many, many cycles in biology. And so a lot of Philip's more recent work here has really focused on some of these kind of cyclical processes, whether that's the cell cycle or supercellular cycles. So as an example here, this is the famous oscillation of gene expression in the root clock, something that determines how the whole organism responds, can respond in a dynamic and environmentally sensitive way, but it was also linking gene expression patterns that were found by doing these amazing isolations of roots and linking them back to the whole organism. So again, very detailed work, but only in the context of the whole organism. And I think that is something that we all are very indebted to Philip for really being a champion of. And that is something carried out throughout developmental biology. So each of these things that were found, these are all important and they're compelling stories, but moving these from the niche of a plant root, something that's of interest to specialists, to something that becomes a textbook example. That's due in large part to really Philip's remarkable ability to present this work in engaging and simple, but not simplistic ways. So this is shortening, good to shorten. Philip's ability to present things really made plant biology and plant development impossible to ignore in the greater developmental community. And what were the sorts of elements of these presentations? He himself was the master of a lot of that, but he also knew the power of very compelling visuals. So to understand something like the development of a root in time and space, you saw a few images of that, but also dynamically. So this is work from a recent paper from his lab, where you understand a lot about the biology going on by this absolutely stunning video where you can see there's movement, there's cells in relation to one another, this sort of idea of how time and space are connected in development. So sometimes these visuals were really detailed and focused on the work at hand, and sometimes these visuals were a bit more metaphorical. So many of you probably remember Philip presenting this. These are murmurations of starlings. The idea that birds, many, many thousands of birds, many, many individuals are flying around together. All together, they create these emergent properties, these movements, these fields. Studying any one of those things individually wouldn't tell you about the system. Studying that, this was kind of a paradigm for systems biology, and a really wonderful visual metaphor. So he had the ability to realize when ideas were very complex and come up with something that enabled many, many people to internalize that and understand the science that he was talking about. Okay, so there's compelling visuals, there's also compelling narrative. So again, we all think about the root, and probably in this audience, we all think about this wonderful idea that the root has radial zones and it's younger at the tip and older at the base, and you can understand all of development by looking at this time. It's time resolved, but in space, and all these things are linked together in this really compelling, beautiful system, which makes you think, why would you ever work on something else? Which is something that some of the people that review my grants sometimes say. But that aside, it's challenged us to come up with ways of saying, well, we can't all study roots. There are some other things to study, but it was such a compelling narrative about what you could study by looking at this organ. Some of you know that I am very proudly now a plant developmental biologist, but I only switched into plants as a postdoc. So I did my PhD in animal axis formation. And at some point I decided to move into plants. And part of that, I was looking back at old notes and realizing that some of Philip's papers are in that collection of kind of coffee stained and photocopied papers of the time that really inspired me to think about working on plants. And so one of them was this paper about root development and mutants, and the idea that you could dissect out some of the genetic components of different tissue layers through these mutants. Another one that was right around the end of my PhD was a review on asymmetric cell divisions, something we still work on. And I was looking at these and trying to figure out, he just had a beautiful way of laying out what were questions that were worth asking, and what were questions that were just about on the cusp of doables, answerable, which is really key. And I will also note that this was not an example of compelling visuals. So this is, I think, the first picture of short root, which it got a lot better after this particular one. But still, so the narrative was the point here. Okay, so amazing presenter, but I also want to mention a couple of other things that become really important for us to be able to do developmental biology, plant developmental biology. And that was that Philip really was an advocate. He had a compelling vision, he could express that, and he could get people to listen and realize that funding plant science was an important thing to do. So he was often chairing panels that were looking at the future of plant biology. He served on the Dev 2 NIH study section, which was the very first grant that I got, was on a session that I'm fairly certain he had to convince other people that plant developmental biology was worth looking at. And also, and you'll hear this from other people, he really was able to speak the language that resonates with many different groups, with academics, with people in government, philanthropies, and venture capital. And then finally, I think Philip's mentorship really produced generations of creative and very community-minded scientists. I'm looking at all of you, not all of you, but several of you over here that really are guiding the way that we do biology today. So I wanted to thank Rachel and Wolfgang for putting this together and allowing us to really think about Philip, or allowing me to think about Philip from this point of view. I just looked at who was presenting, and I'm sure that I've missed some, so I apologize for that, but look at how many people are presenting. This is just the talks. This isn't even the posters at this meeting, who were the trainees or the grandchildren, the trainees of trainees from Philip, and it's really remarkable. And then finally, I think a lot about what Philip did in combining time and space in development, and so I want to call out some people that I've really enjoyed getting a chance to interact with over many years. So in time, Kim and Ken started their labs around the same time as I did, and we've gone through many things together. And then in terms of space, Siobhan, who's close by at a different university, and Jose, who's literally across the hall. It's been really inspirational to work alongside of them. So I want to kind of end, just want to finish with an appreciation of the ideas and the work and the actions of Philip. This is actually an image of him from an event at the GMI in Vienna, and it actually reminded me of another conference that was also in Vienna at the very beginning of my career. And it may have been the first time that I'd met Philip in person. And it remains one of the most bizarre conferences that I've ever attended, and included some fairly sketchy, not legal transportation, a hotel that we suspected very strongly was haunted, an evening event where after about an hour of electric punk violin, Philip staged an intervention, and he started a revolution, and essentially we all left and into the dark, deserted back alleys of Vienna in February, which meant, but we all followed, and I think that means that with Philip, we knew you were both in capable hands and in for an adventure. So thank you, Philip.
The next speaker is Joe Ecker from the Salk Institute, talking about Philip's impact on Arabidopsis research. Take it away.
Thanks for the opportunity, Rachel and Wolfgang, to talk about my colleague and friend, Philip. That was really nice, Dominique, really an amazing tribute. I want to take a little bit of a different approach. I am not a developmental biologist. I've never claimed to be a developmental biologist, but Philip and I got along very well as we were both interested in technology. And let's see, what do I do here? Pointer, forward. Okay, so Philip sort of fell into the category of what I would say a developmental biologist who was also a technology developer. I really appreciated how that technology would allow you to answer new questions in biology. So I went over the 200 plus papers that Philip has published and picked out some of my favorites to illustrate how he incorporated technology into the science that he gave us. And so I just start back from the time when I first met Philip when he was a postdoctoral fellow in Namhai Chua's lab. And the technology here was plant transformation, if you could believe. This was the early days. Here we're in 1989. He was using plant transformation of petunia or other plants, tobacco, to dissect the regulatory elements of what people were using as a constitutive promoter, the 35 cauliflower mosaic 35S promoter. And Philip nicely showed, oops, I gotta go back here, that this promoter could be dissected into A and B elements, each of which caused the expression of that, the gene attached to it, it was probably Gus or something in this case, to be expressed in a different way. So A was expressed in seedlings in the root, B was expressed in vascular tissue in the roots and hypocotyl, and combined they were in all cells. So he first demonstrated really in a series of about 5 papers of continuing to further dissect this promoter. and the technology was. And I actually picked out the same paper because the technology here is genetics, right? Doing a genetic screen, a large-scale genetic screen at that time, it was early days for Arabidopsis to start plating out and screening for mutants. And he identified several of the key mutants that his laboratory worked on for the following decades. And that also, this was this collaboration. So Philip had, you know, it was hard to kind of view at the time whether they were collaborators or competitors. It turns out, you know, it was sort of both. They worked together very well, had their, you know, were working down the same question, but yet were able to collaborate. So Ben and Philip had a number of papers together, and also John Scheffelbein, you know, his work on root hairs, et cetera. So these were some of the attributes. Then, you know, what was the technology here? The technology was to screen tDNA lines. So this was, what was the year here? 1996. There was no Salk collection, right? There was no sale, there was no Gavi. Ken had developed a population at DuPont, and they did a screen and identified, and of course, Philip was really a developmental geneticist with the emphasis on starting out as a geneticist. He had 2 alleles that he identified in the scarecrow gene, which, you know, then he began to characterize. So the technology there was, you know, insertional mutagenesis, that he was an early adopter of that. Then I'll point to Jocelyn's paper here, where, you know, a lot of the beautiful images that Dominique just showed are due to hard work in identifying specific promoters expressed in different layers of the root, in the epidermis, the cortex, pericycle, et cetera. And so the technology here was to develop a set of promoter fusions that allowed then later additional studies of those cell populations, because now you could tag them. And so this was also another one of the key genes. I won't go into the details because Dominique has showed these pictures already. But I think there's a beautiful, couldn't put it here, but there was a beautiful animation that Philip shows. If you're really interested and this is new to you, go watch the iBio 2 talks that Philip gave about root development, and you'll see some of these beautiful animated images that he showed of short root moving from the pericycle, from the here to steel into the endodermis. Beautiful work. Yeah, so this is the classic image that everybody shows, and Philip gives a beautiful lecture about that. So once you have all of those cell lineage markers, what can you do with them? Well, what Philip initially did, and this was Ken's work that was mentioned, is to use those lines to do fact sorting. And probably the only person in the world, this is probably not true, but David Galbraith is a real expert in fact sorting. So I know that Ken and Philip got a lot of pushback about, can you use protoplasts for these kinds of studies? And Philip was smart enough to know that you could actually produce the protoplasts, identify a set of genes that maybe rapidly respond, and just eliminate them. And so he used the Affymetrix chip, Ken, to sort these cells and make the first sort of gene expression root map for Arabidopsis. So the technology here was use the lines that you had developed, you know, for cell lineage, and couple that with fact sorting, and then use the latest technology at the time, which was Affymetrix chips. And then Shahan later extended that, instead of just looking at the root tip, to adding a sort of 4th dimension, the time dimension, which is really interesting, and also applying that same technology to get a little bit more information about the so he didn't stop at gene expression. He said, hey, we got to look at proteins. Okay, so we now have a protein expression map that was produced and published in PNAS. Again, using the same tools that were developed over the decades. And this is the beautiful, I really love this sort of oscillation story here, where, you know, this is a fusion of DR5 actually to luciferase, and the identification that you can have these sort of, Philip related this to somite development, that beautiful work that had been done in looking at a clock that partition cells, cell groups into somites. And this was the sort of beginning of a study which was continued. I'll show the continuation about how to follow this story. This is an interesting phenomenon, but what's the cause of this? And so other technologies, and I mentioned this, Wolfgang Busch is my colleague at the Salk, were things like root arrays, just keep pushing the technology. And I won't talk about his, outside of Arabidopsis, he worked on grasses, he worked on, rice. He had done imaging using different technologies to image roots in pots and things like that. They're really amazing. So he collaborated with engineers, mathematicians, et cetera. In fact, I went and reviewed, he had a systems biology center funded by NIH, actually I think it was probably the Cancer Institute, that involved a lot of folks. And so he brought a community of people together after he stepped down as chair, took on another role to build a systems biology center, which I think was really super. I happened to be on the review committee, and everybody unanimously thought it was a fantastic center. Yes, so the continuation of that story, looking at the oscillations of the clock, was this really beautiful story by Janice Dickinson. And what's the technology here? The technology is biochemistry, chemistry. Not to leave it, there were other earlier papers suggesting there was a carotenoid that was involved, but actually identifying not only the molecule that was important for this oscillation, but also identifying an Arabidopsis mutant that reduced this, blocked this process. So again, utilizing these tools, continuing to push the biology using technology. And I really like, there's a series of papers, I can't actually, Dominique showed a list of them, that I think are really super interesting. And Philip had been doing this from the beginning, as was said. He was sorting cells. We had a collaborative paper, so I had that opportunity to collaborate with Philip and Natalie, who's here, where we sorted cells. Philip's lab sorted cells, and we sort of parsed them out. Natalie looked at microRNAs, and we carried out DNA methylation studies in those cell populations. And that was a really fantastic collaboration. But as was mentioned, this was a decade earlier than these studies, which now take it to a whole different level. Really one of the first sort of detailed papers of the root using single cell technology. And there are many others, which I don't have time to talk about. So this is one of the latest papers, and Dominique showed a video, I believe, of this sort of process here. This is the 4D. So this was really recent, actually after Philip passed away. And the reason I show this is there's some cool, so Philip said, hey, we need to figure out a way to do this time dimension in real time. And the people that have done this that are in the audience can tell you more. But he worked with Scott Fraser, who's a master microscopist, to build a laser scanning confocal microscope that would allow you to do live imaging over time. I think over a few days, every 15 minutes would take a picture. And the reason that I show this is in part that this statement here refutes an earlier paper from Philip's lab. That is, there was a model of bistability that in terms of short root and scarecrow and their presence in cells that you would have a switch. And this paper, the title here says long-term, which reveals the dynamics inconsistent with bistability. So don't hold on to your hypothesis about what happened when you have new data that says that wasn't right. And a lot of people wouldn't necessarily, they might, well, we were partially right. No, this is a clear statement that says we actually now were able to measure quantitatively the amount of scarecrow and short root and which amount together triggers cell division and partitioning of the cell. So this is really, I think, something to think about in terms of your own science is that, science moves on, new data emerges and models change. So the last thing that I will mention is that Philip was, all the attributes that Dominique said, or I just can't reiterate that we worked together on many of these, projects, convincing NSF, and he was the most eloquent speaker to the government. He knew, like you say, he knew the lingo. He was able to get, push projects that were not necessarily in his own interest, but really good science, and convince agencies like NIH that they needed to consider plant biologists, developmental biologists very seriously, because this is, really important science. So I just want to point to just one event that we had together with Philip and his longtime collaborator, Gloria Caruzzi here, and one of our keynote speakers tonight. So I'll stop there. Thanks very much.
Thanks, Joe. The next speaker will be Uwe Ohler from the Max Delbrück Center in the Humboldt University in Berlin. And he has been a partner in crime of Philip in terms of systems biology. So.
So thank you, Rachel and Wolfgang, also for giving me this opportunity to reflect and talk a little bit about the work that we did together with Philip in the context of systems biology. Philip moved to Duke after the attacks on 9/11 to take over the chair of the newly merged biology department, and very shortly after he started putting together a biological networks reading group, actually, in the very beginning. And so this was a very self-organized effort to bring together people that were interested in this, at the time, new field of systems biology. So to try to quantitatively describe biological phenomena, but at very, very different scales. And over the years, with a lot of perseverance and with a lot of patience, it turned into the center that Joe just actually mentioned. We had an NIH-funded center at Duke that combined almost 20 faculty across many, many different disciplines. So on the quantitative side, it also includes many different areas. It was math and stats, but also physics and computer science and engineering, and these are all very different. And Philip actually knew that these were very different and that he needed different partners to address different questions and to work with different technologies. And I mentioned the patience because I remember one very early meeting that we had when I almost lost it after, one of my dear colleagues from physics asked, it's like, it's maybe the 5th time that I'm asking this, but can you again explain to me what a transcription factor actually is? And I'm like, really? Just take a textbook, read it. And Philip would just have this patience and really try to open up this field, not just roots as a system or plants as a system, as was mentioned, but also biology as a whole to quantitative researchers. And so the center itself actually had many different areas. It focused on, not surprisingly, actually somehow on all different areas that Philip himself was interested in, right? So there's definitely development was mentioned a lot. There's also, of course, cellular networks that was mentioned, stress responses, right? There was also abiotic stress responses in the root, maybe evolution a little bit less. And in terms of all these different areas that he was interested in, like gene expression or studying oscillations, as we just heard, or looking at the topology of different root patterns in different species, or then branching out to synthetic biology in the companies, you need different researchers to work together. And Philip really came with this appreciation that people could bring stuff to the table that could work well together and where people together achieve more than the individual parts. And in addition to the center as a whole, this really goes also in the individual collaborations. All of these papers addressed very different biological questions that had a quantitative angle and where he collaborated with different quantitative researchers that brought a different toolbox to the table. I came to Duke in 2005, so 2-3 years after he started there. And sort of my impression is mirrored actually one of my students later on that also met him during the interviews, right? So I also met him during my interviews there first. And then after I joined, it actually didn't take too long for me to join the systems biology reading group, and for a student, Juliette Colinas at the time, to bring us together. And so the first 2 papers actually that came out of my own lab within a year. So these papers were published within like 12 or 15 months actually of me starting at Duke. We had 2 publications that I think really symbolizes this tight connection, that you can have work that isn't service to one another or helping each other out, as valuable as that is, but rather to pose different problems to each other so that each of a collaborating team has something unique to work on and to contribute to a larger problem. And so here we were very early on in quantifying these beautiful images that we saw and reading out the cell type specific spatiotemporal expression patterns out of very small image data sets at the time. And that was sort of the beginning of us working together. And over the years, actually, this included this amazing group of people from my lab and from Philip's lab, a lot of them roughly in this direction. And to come to this stunning number of 15 publications that we jointly had, many of them actually shared in various configurations, 2 of them actually still being in the works right now. And I was thinking, like, I don't know many cases where so many publications result, other than maybe you have a lab technician job somewhere, or maybe you're married, right, and you run a lab together or something. And it's just been a real privilege to be part of this. And so I just want to show a couple of examples here. One was mentioned, where I think what Joe also mentioned really comes to fruition. This idea to use the latest technology and define massively challenging problems to your students and postdocs, but then also support them of getting these crazy ideas done and having long-term vision to promote this, right? The Root Atlas started with Ken and continued with Rachel, and this is a project over 20 years, essentially. And so we got into this in the collaboration with Shahan at the time at Duke. And I bring this as example because spatial omics is like the hot shit right now. Everywhere you hear spatial omics. And what Shahan did at the time to actually dissect single roots, fact sort them, and get these spatiotemporal expression profiles more or less was 15 years ahead of a resolution that later could be achieved with Visium 10x commercially. It's 15 years ahead, and this is just stunning to me. And then it continued with probably the craziest project I ever was involved in, and that is the root array that Wolfgang sort of took over eventually and made it come to life and to work. And again there, this was enough material, so to say, for us as a lab to work in, that I had 3 students actually contribute individual methods to this massive project, from trying to track plants as they are growing, to segment them out, to read out the expression values in this, all in 3D. The problem turned out to be much harder than Philip made it sound in the beginning. Maybe that's also a common topic, but then you were hooked, and then you had to keep going because you wanted to know how the story ends. And so the root array was one of these crazy projects, and you can just see here how challenging this was. This again is over 10 years ago, and now again imaging methods get to the point of having other systems to catch up with plants to finally be able to do similar things and achieve similar resolution like 10, 15 years ago. And of course, I couldn't leave off by not mentioning of the single cell work that we were able to do together recently. We had a shared student, Chewei is actually here, and you can hear much more about how we use this data in the context of systems biology to, as a foundation for generative AI models that can now simulate single cell expression patterns and the development from the quiescent center in different mutants. And so that's really cool work, and Chewei will talk tomorrow about this in the single cell session. And finally, Wolfgang, here's the book, the slide that you were looking for. Because I had a similar experience. After I moved to Berlin about 10 years ago, I noticed that Philip was visiting quite frequently. And by chance, I also walked into a bookstore and saw this book by his brother, Christopher, that tells the story of the family. And there's an amazing story to be found here, and I'm really not the right person to mention much of this, other than that I was able to convince Philip to talk about one aspect, because just before the pandemic was the 100th anniversary of Bauhaus, of this famous German architecture and art university, and his great uncle and aunt were actually teachers of Bauhaus, and he was able to present and tell us a little bit about this family story. And in fact, I was able to accompany him and one of his cousins who came to visit to Dessau to actually visit the university and visit the house that his great uncle lived in, and so get to see a little bit of this context that I guess made Philip to this amazing human being that we all appreciated. And with this, I just want to finish with a couple of statements that the people from my lab actually just wrote back as a first response after they heard the news that Philip passed away. And with Philip's last email, actually, just a few days before he passed away when he came back from the hospital, because we were still working together on this AI generative model for root development. Thank you, and thanks again for giving me this opportunity.
Thank you, Uwe. So the next talk will switch gears, and we will actually hear something about another facet of Philip, the entrepreneurial Philip. And Natalie Brakefield from New Leaf Symbiontics joins us and will talk about this. Thank you.
Okay, thanks everybody. Thanks Wolfgang and Rachel for the invitation to be here. I wouldn't say I'm happy to be here, but I'm very much honored to be here to talk to you all about how Philip was an entrepreneur. So why am I the person here today to talk about this? He was my PhD advisor at Duke University, and I was a non-traditional student. I showed up with 3 kids in tow. They were ages 6, 9, and 10 when I showed up at Duke. And I was just walked right in and said, "Hey, Philip, do you think I can join your lab?" And today it maybe wouldn't be such a big deal, but 20 years ago this was a little unusual. And so he said, "Well, let's see what you can do." And he gave me a chance, and I was able to join the lab and, of course, work on some really amazing science, meet amazing people, and my career wouldn't be the same without this. So when I graduated, he said, "Hey, are you interested in going to industry?" I said, "Oh, absolutely not. I'm very much going to academia." I did a really very lovely postdoc, and then I went to a startup. I went to New Leaf Symbiotics in St. Louis, Missouri. It was just an opportunity I couldn't pass up. But the things I learned in his lab, I was always able to use in my job in industry. And actually, I talked to Philip's entrepreneurship class every year for the last 5 years. And in fact, some of the last times I interacted with him and talked to him were in September, right before he passed. He missed the class that I was going to talk at. He was actually in the hospital. And so, of course, no problem. We can still do the class without him. And I still talked to his students, and everything went quite well. And I reached out to him, how's it going? And he said, "Oh, no, everything's just fine. I'm just a little tired." And then that was less than a week before he passed. So, of course, what I'm here to talk to you today is about Philip the entrepreneur. And as I was sitting here thinking about it, I wanted to think about what are some of the things. Now, there's a lot of other things beyond this list, but what are some of the things that a startup needs to be successful? I think you really need a visionary leader. You have to have somebody that can understand the market, can understand the conditions, can attract the key employees, can raise the money, like talk to venture capital people. A lot of people have mentioned that, how he was such a great communicator. You also need a good idea. You need good technology. It needs to solve a problem. And he was very good at identifying what kind of technology was out there, what was he developing, what was being developed around him that could be used by industry and would be valuable to someone. And so this is my attempt at starting to make a network diagram. So he was involved with 3 different startups. Grassroots Biotechnology was founded in 2007 with Doug Eisner, and so the technology was root array. We've seen this already a couple of times. They also had novel genetic elements that were, then this company was acquired by Monsanto in 2013, which then itself was acquired by Bayer in 2018. The second startup was called High Fidelity Genetics, High Fidelity Technologies, founded in 2014 with Spencer Wan. The technology was root tracker technology, so it was monitoring, it was sensors in the soil to monitor how roots were developing out in fields, and they had some breeding methods for improving crop yields. And so they had some really nice technology there too. The most recent startup, and so actually I know he was working on this right up to his passing, is Raleigh Biosciences, founded last year with Ross Hassani. And the technology is a lot of single cell technologies, and there's some really cool 3D bioprinting of plant cells. And so he's, I know Ross is still working in this startup and trying to get it going. And so in all of these companies, Philip was really this visionary leader that someone needs to be able to bring people together and make your startup successful. So as I was also thinking about this, what were some of the things I've learned from Philip? We've heard some of these before already. Help others. It's good to collaborate with people, try to collaborate, maybe not so much compete, but help each other. Perfect is the enemy of the good. Just get to the next stage. Boy, that was a hard lesson to learn. I always wanted one more really nice figure. Just need to do one more experiment, and it's gonna just be so much prettier. He's like, oh my gosh, just do it. Get it published. Go after the low-hanging fruit. I know you guys have all heard him say that. Low-hanging fruit first. Don't go after the most difficult problem right away. Like, go after that. And this one made me laugh as I was making it. Never use more than 10 words on a slide. I was like, oh. So as I was making the slide, I kept revising it. I'm like, 40, close enough. So, so as I moved to this, I think about what makes life successful. You know, there's no way I'll be able to read this to you, by the way. So I'll just point out a few of the lines. This is a slightly altered version of this poem. He has achieved success who has lived well, laughed often, and loved much. I added made the world better with an improved root, and whose life was an inspiration. And so I just want to close by saying I feel very fortunate to be part of the Benfey Lab family. And again, I'm really grateful that we had the opportunity to tell him how much we benefited from him while he was still here to hear it. So this was the Benfey Lab Symposium at his 65th birthday in summer of 2018. So thank you very much.
In our last few minutes, we will now hear about Philip as a mentor. So we'll start with 2 recorded, very brief tributes, and then continue with several of our speakers who are here in person. The first is Siobhan Brady's recording.
My name is Siobhan Brady, and I was a postdoc with Philip Benfey at Duke University in the years 2005 to 2008. I first met Philip at an ICAR meeting in Madison, Wisconsin around 2003. I was already aware of his work about short root as a mobile transcription factor and understood how it was a paradigm for how plants signal in terms of developmental biology. At that meeting, he presented he and Ken Birnbaum's work on cell type resolution microarray profiling. I was completely hooked. At the time of my postdoc, he had assembled what was a dream team of postdocs and graduate students. People who were ambitious, incredibly intelligent, supportive, and constructively critical. One of the really important lessons that Philip taught me was about strategy, how to embrace thinking outside of the box and how to pursue questions of broad biological importance. Possibly the most important lesson Philip taught me was how to be an excellent mentor. Philip was one of the most important people in my life. I am forever grateful to him, and I miss him greatly.
My name is Jiyoung Lee. I was a postdoc at the Benfey lab from 2003 to 2007, and I'm currently a professor at Seoul National University. I was extremely fortunate to be part of the Benfey lab and to participate in the cell type specific root expression map project with great colleagues. Philip's advice and support helped me build my research program as an independent scientist throughout my postdoctoral training and beyond. For a female scientist from South Korea, where advocating for female professionals was rare at the time, Philip's support was very special to me. Attending Philip's memorial at Duke University last April, I learned more about him not only as a great scientist, but also as a person full of humor and love. Philip's outstanding mentorship stemmed from his genuine sincerity toward his people. Philip Benfey's legacy will continue through the mentoring of his successful mentees around the world. Nevertheless, will be missed so much, Philip.
Is Uka here? Oh, there, Uka.
Yeah, my name is Ecker. I represent the New York University era of Philip. The group was still relatively small at that moment, and it was kind of interesting to see how this group started to develop towards a stronger unit. So Philip definitely was a great role model for me in establishing a group at that moment. I just want to also establish one thing, kind of concentrate something that something else. So the idea that he toured 6 years the world before he actually committed to science. So first, I was initially kind of impressed by the spatial aspect of it. So he was often mentioning the Trans-Siberian Railroad. So as a Finn, I was kind of amazed because I never actually even went to Russia myself. But more recently, I have a daughter who just graduated from the school who doesn't really know what to do. And I feel kind of sure that you can take your time, so that you can take your time to figure out what you want. Time is on your side. Thanks.
Next, we have Terry Long, who's from North Carolina State University.
I was a postdoc in Philip Benfey's lab from 2005 until 2010, and it was one of the best times of my life, and partly because with very little effort, Philip was able to create a work environment and a learning and research environment that was very collegial, yet very scientifically stimulating. Many of the people that you see on this side here are really good friends who are former members of his lab. Here, we're in Japan at ICAR in 2008. And like Ken asked for all the postdocs of Philip's lab to come together, and I didn't realize how many there were. We came out in droves, and we had an amazing time. So much so that even now, we're in group chats together, and when we have problems, we'll go to each other and we'll say, what should we do? And we would ask each other, well, what would Philip do? So I would call him every once in a while for career advice, and he would always pick the phone up no matter what he was doing. And that showed me that he was an amazing mentor. And sometimes I would see him at various conferences, and he would always, hey, Terry, give me this warm hug. And so I began to think of him as a dear friend who will be sorely missed.
Next, we have Jamie Van Norman, who is joining me as of next week at UCLA.
I did not make a slide. So I came to Philip's lab as a sort of unusual trainee. I wasn't interested in transcription factors. I know, it was very painful for everyone. But I was interested in roots and how root cells coordinate developmental activities. So in his lab, I really had the space and the opportunity to grow as a scientist and as a person. I don't think there's anywhere else where I would have had the opportunity to travel and speak at so many conferences and meet so many people. And this really opened a lot of doors for me at UC Riverside, where I eventually started my own lab. It's really hard to know that Philip isn't just a phone call away. Like, we could just call him. But I think of him often, and I strive to be a mentor and a scholar and a scientist in a way that would make him proud.
Our next speaker is Jazz Dickinson, who's an assistant professor here at UCSD.
I want to thank Rachel and Wolfgang for the honor of being able to speak on behalf of Philip's many mentees. You saw that picture, and that was only some of them, right? There are so many people who are now successful in academia, industry, and other professions. And there's so much to say about Philip as a mentor. I really struggled to narrow it down. I used to have a motto: Philip is always right. Because he just was. If he gave you scientific advice, you should take it. And now I find myself asking WWPD, what would Philip do? He often described visualizing science as a game of strategy, like chess. And he was really good at seeing the whole board. He didn't get lost in some detail on the side. And he was almost clairvoyant at times with his uncanny ability to figure out which experimental pawn was about to be promoted to a queen. So I just want to say thank you, Philip, for all that you taught me. You will be deeply missed.
Next, we have Trevor Nolan, who in 2 weeks will start his own lab at Caltech.
I've had really the honor of spending the last 5 years working with Philip and the wonderful people in his lab. And when I think of Philip, I think of cutting edge. I also think cool. Like, Philip could do things that nobody else could do, like theme his talk about roots around Formula One racing. So I still remember riding in his BMW and him showing me how it made a hologram of the BMW symbol on the ground when you opened the door. And I remember starting, wanting to do single cell analysis, and this is when people were doing 1 or 2 samples, and Philip said, let's do time courses. And I was hooked. And we had the most wonderful time. And I still remember the grin on his face when he read aloud the reviewer comments that said, it would be frivolous to ask for additional experiments. It's been such a joy working with Philip. We're going to miss him dearly. I still think about him daily and think about this wonderful community that he's generated.
Next speaker is Pablo Sekeli, who is currently a postdoc at Duke in Philip's lab.
Thank you. Yeah, I want to share a personal story. So I joined the lab in 2020, just before the pandemic. But even like before, after I got accepted, before joining, Philip told me, we would like to use your help. You have unique skills. We have this small project you need to push. It will take you 2-3 months, and then you can start your own thing. And I joined the lab, and things didn't go as we planned. And these 2-3 months became 2 to 3 years. All the time we had different problems, and we had to change perspectives and rethink of what we're going to do and how, and we always had to come with creative solutions. And during all of that time, Philip was so patient, so optimistic, always wanted to help and like took his patience. And at the end of the day, all of that work wouldn't have worked. And clearly, he really wanted this paper out from the get-go. And yet, he was a great, great mentor for me, and I will miss him dearly. Thank you so much.
Come on up, Mingji. You don't have to wait. Next, we have Mingji He, who is currently a postdoc, also currently a postdoc in Philip's lab at Duke.
First of all, thank you, Rachel and Wolfgang, for this opportunity to express my appreciation to Philip. So Philip has been an extraordinary mentor and inspiration to his trainees. His unwavering encouragement and support have been the cornerstone of my growth. And beyond sharing his profound knowledge, Philip invested time and provided guidance with remarkable patience. He respected and valued each single trainee and recognizing our unique strengths and potential. His belief in us was so powerful and encouraged me to explore, learn, and innovate. Philip's open door policy and the emphasis on critical thinking have profoundly shaped my approach to the scientific inquiry. His dedication to nurturing young scientists made him an invaluable asset to our young scientific community. I deeply appreciate his mentorship and the lasting impact he has had on my personal and professional development. Thank you, Philip, and thank you, everyone.
Finally, to close, our last speaker is Mingyuan Zhu, who is also still a postdoc in Philip's lab at Duke.
So good afternoon, everyone, and thank you to Rachel and Wolfgang for giving me this opportunity. So my name is Mingyuan Zhu. I'm a postdoctoral trainee of Philip. I joined his lab in August 2020. My research focused on the adaptation of rice roots to soil stresses with single-cell techniques, with Philip's generous and invaluable guidance and support. So today I would like to share a memorable moment. This is a photo taken during one department seminar. So in this photo, you can see Philip dressed in a suit stood next to me throughout the event. So in this photo showed two of my strongest impressions of Philip. First, being elegant. So it actually also nicely presented with all the photos shown in this session. And Philip is always well-dressed and carried himself with a distinctive and inspiring presence. And the second is his continuous support. So Philip actually encouraged and backed almost everything we like to try. So I feel incredibly fortunate to have the privilege of being one member of Benfey Lab. And thank you, Philip, and thank you for listening.
With that, thank you so much for your attention and for coming. Thank you for your patience while we ran a few minutes over. We really appreciate it. Thank you, Wolfgang.
Thanks, Rachel, and let's thank all the speakers again for their contributions for the memo.